Hello, everyone. Thank you for joining us today for this webinar, Comprehensive Genetic Characterization in QC of Genetically Modified Cell Lines. My name is Charlie Brault, and I'll be your moderator for today's session. It's a pleasure to introduce our speaker today, Irina Sergeeva. Irina received her Ph.D. in molecular biology at the University of Amsterdam, where she studied the mechanisms of cardiac hypertrophy and repair. Irina generated several transgenic mouse reporter lines for in vitro and in vivo analysis and used 4C sequencing technology to study gene regulation in the heart. Irina transitioned to industry 10 years ago and started working for Syngenta, now Solvius NL, as a product developer and research technician, where she performed targeted locus amplification assay on customer samples and practiced analyses of NGS data. Currently, Irina serves as a scientific account manager at Solvius, managing sequencing projects for the genetic characterization of modified cell lines, animal models, and cell and gene therapy products, supporting IND and BLA submissions. In addition, Irina leads a team of scientists responsible for the analytical processing of the sequenced data. Throughout the webinar, you can submit your questions at any time using the chat box on the left of your screen. We'll collect them and address as many as possible. With that, let's get started. Enjoy today's presentation.
In this presentation, I will discuss how genetic characterization and quality control of the genetically modified cell lines is currently performed at Solvius. Solvius is a CDMO which consists of several sites located both in Europe and in the United States. At Solvius, we perform the analytical testing of different drug products at various stages of their development to ensure the drug's safety and efficiency. In Utrecht, the Netherlands site, we focus on genetic QC solutions with more than a decade of experience in the cell line development field. Why and when the quality control must be done at the genetic level? Quality control is obviously important at the late stage when you want to file your master cell bank to the authorities, but not only, it's also important throughout the entire process. If you think of a vector that you will be using for your cell line, then you need to check that the vector is correct and has all components you expect. Then, when you test your transfection method, you also want to be sure that the vector is integrated and the method is efficient. Later on, you get a lot of clones which you need to screen and select the best one which produces the biologics. After the clone is selected for cell banking, the master cell bank has to be thoroughly characterized. The characterization is regulated by the authorities, which issue specific guidelines. For example, ICH Q5B guideline is mostly used in this case. The guidelines list the criteria, including the vector integration sites, vector copy number, and vector integrity. Additionally, at this stage, monoclonality and genetic stability are important characteristics of the cell bank because they also ensure the high quality and safety of the final product. This presentation aims to show which analyses are done at different stages. As I have mentioned, one of the characteristics of a cell line is vector integration sites. At the moment, integration sites can be detected with different techniques. Some, but not all, are listed here. At Solvius, we use TLA, which stands for Targeted Locus Amplification. TLA is an unbiased method in integration site detection because we only need a sequence information. And that is in contrast to some targeted approaches like targeted Oxford nanopore I will discuss later. TLA data has nucleotide level resolution in contrast to FISH, for example. With TLA, we sequence not only integration sites, but also the vector sequence itself at the same time and in the same experiment. That allows us to characterize the vector integrity, including the sequence variants and indels, structural rearrangements, concatemerization. Unfortunately, copy number estimation is not that strong with TLA. Some other techniques perform better here, like ddPCR, and I will also talk about that. And if you want to fully characterize some complex integration events, TLA can be complemented with other techniques using long-read sequencing. As you can see, a lot of characteristics can be covered with TLA, and it would be indeed great to have one covers all technology. However, we do use a combination of technologies that gives a more comprehensive and sometimes more accurate picture. But let's first dive into the technologies themselves. I will talk about TLA, ddPCR, and targeted nanopore sequencing, also known as Oxford nanopore technology. We use these assays routinely. At the same time, our team keeps developing and implementing other NGS-based assays like capture hybridization, TLA-based capture, long-read whole genome sequencing, and others. TLA stands for Targeted Locus Amplification. In TLA, we cross-link DNA in its natural state in the live cells. After that, DNA is fragmented and ligated to generate DNA circles. These circles consist of reshuffled DNA fragments from different parts of the chromosomes. Each cell will produce a different set of circles because of stochastic reshuffling. After that, inverse PCR is used with the primers designed on the vector. The integrated vector serves as a viewpoint. After PCR, only selective set of circles is amplified. Those enriched circles contain not only the vector DNA, but also the genomic DNA surrounding the integration sites. The enriched product is then sequenced and mapped to the host genome and to the vector, producing sequencing coverage. This is a schematic of the coverage across a genomic locus where a vector is integrated. The highest coverage is observed at the point of integration, and it decreases on the flanking regions. Using these reads, we perform further analysis and compile the data in a report. Let's now go to a real example. We have recently collaborated with the NEAST consortium, which aims to compare and standardize different methodologies in different processes, including genetic characterization of cell lines. We and other laboratories have received 5 cell lines, each containing from 0 to 4 copies of a construct with GFP. We performed TLA on these cell lines and detected nice coverage peaks on the genome. These are the integration sites. For example, for sample VCN4, which contains 4 vector copies in black, you would see 4 peaks on the genome indicating 4 integration sites. In the report, we show the zoomed-in coverage of each integration site. For example, for chromosome 16, the vector is integrated at the place where the highest coverage is observed. We also report the coordinates of integration on the chromosome and on the vector side, and provide the sequences of those flanking regions. What is interesting to note here is the peak formation in TLA. In the original publication describing the same cell lines, it was indicated that for the VCN4 sample, one of the integration sites was on chromosome 4, while with TLA and also with other techniques performed by the other labs, chromosome 4 integration site was not observed. Instead, an integration site on chromosome 16 is reported. Why is that difference? I have just shown you the peak of coverage at the integration site on chromosome 16, but on chromosome 4 there is no or just background coverage observed, indicating there is really no integration here. Apparently, the genomic sequences surrounding the vector integration site are homologous to multiple regions in the genome, so they are present in the genome several times, and one of them being on chromosome 4. Without extra evidence, the integration is randomly assigned to another chromosome, like chromosome 4, although that is a false positive call. So TLA does not rely only on the breakpoint sequences, but it always takes into account the accumulated coverage around the integration sites. Using the TLA sequencing data, we don't only look at the integration sites, but, as I said previously, we describe the vector integrity. By comparing all the obtained sequencing reads with the reference vector sequence, small variants and indels are called. Standardly, we report the small variants which are found in 5% of the reads, but we can go as low as 1% in some cases. To do that, we aim to obtain really high coverage on the region of interest to be sure that low-frequency calls are reliably called. To illustrate the detection of the sequence variants, we called the variants in the same cell lines with the integrated GFP copies. And we could detect several mutations at different frequencies. We report the expected position, the identified mutation at this position, coverage, and the observed mutation frequency in the data of 2 primer sets per 1 sample. In red, the mutations are highlighted which are found in the individual cell lines. Typically, TLA does not provide absolute frequency of the mutations, but in these cells, the calculated frequency was quite accurate, meaning that in VCN2 cells containing 2 copies of the vector, it looks like only 1 copy had the mutation, because the frequency is approximately 50%. And in the VCN4 sample, the sample with 4 copies, the frequency is about 20-25%, indicating that 1 out of 4 copies had the mutation. The percentages can be guiding, but are not always precise in TLA data. Using the same sequencing data, we can see not only mutations in the vector, but also bigger structural changes detected as so-called fusion reads. The fusion reads consist of 2 parts of the vector which were not originally close to each other in the vector, but now were observed in the same sequencing reads. These reads can indicate deletions in the vector copy, or more often represent connections of several vector copies, or so-called vector concatemerization. Because we use short read sequencing, we only see the connections between the copies. We report the positions at the fusion points and conclude that a vector concatemer has integrated into the genome. Further reconstruction based on this data is typically not possible, but remember this slide, I will come back to that a bit later. I have shown you the detection of sequence and structural variants in the vector, but using TLA data we can also detect even bigger changes on the genome in the region of the integration sites, based on the assumption that expected coverage on the genome should look like a peak, where the vector is integrated in the middle. Often, we see deviations from this expected coverage, and that indicates that some genomic changes have happened. Either they were already present in the cell line genome, or they have happened during the vector integration, or maybe even after some prolonged culturing. If the genome is unstable, these changes can be detected. These are the simplified schematic pictures, but they illustrate that we can detect inversions, deletions, genomic duplications, and even connections between different chromosomes, or so-called translocations. In a real example, using VCN3 sample with 3 GFP copies, one of the integration sites showed a gap in coverage next to the high peak. The peak is where the vector is integrated, but next to it is a 648 deletion, which is also simplified in a scheme of integration in our report. The scheme is not up to scale, but it summarizes the finding of the coverage profile. Copy number estimation can also be done with TLA. Copy number is calculated as a ratio of coverage on the vector and on the genome, and here I will explain how. Here, the vector reads are shown in red and the genome reads are in blue. If two vector copies are integrated, the actual coverage distribution looks like this, and at the integration site you expect to see approximately the same number of reads on the vector and on the genome. However, when the data is mapped to a single copy of the reference vector sequence, the total number of reads on the vector is actually higher than the coverage on the genome. This ratio of coverage on the vector to the coverage on the genome at the breakpoint gives the number of 2, meaning that 2 copies of the vector have integrated. Unfortunately, these numbers are not always so precise and neat with TLA, especially when the copy number is high. So TLA copy number is then just a rough estimation, and that's why we at some point had decided to switch to ddPCR copy number calculation. As an example, again, in the same cell lines with 1 to 4 copies of GFP, the assay was performed on the RRE vector element and using the RPP30 gene as a reference. After normalization, 1 up to 4 copies of the vector were detected in these cell lines. I would like to note that genomic reference is often and commonly used for normalization, but it's not always the best option because some genomes might be unstable. The calculation of copy number is based on the assumption that your cell line is diploid, which is not always the case, or diploid is not known for some cell lines. Then we can implement another approach. We do use the integration site breakpoints identified with TLA. Those sequences will be present in your cell line only once, and then they can be used as a solid reference for the copy number calculation. Another interesting fact is that if with ddPCR you identify 4 copies in 2 different samples, it doesn't mean that the samples are the same, because one integration site seen with TLA as one peak might have 4 vector copies at one location, integrated as a concatemer, and it will look the same as 4 integration sites with 1 vector copy in each site. The message here is that copy number estimation, the same as gene expression, is not sufficient characteristic by itself to conclude that the cell bank, 2 cell banks have the same or different origin. By now I have covered TLA and ddPCR, and now I will show how Oxford nanopore technology can complement this data. In many cases, integration sites might look a bit more complex than others. For example, the cell line DP12, based on CHO cells and producing antibodies, show this interesting pattern of vector integration in TLA sequencing. It has spikes, small peaks, wide broad peaks, so none of them look like expected TLA coverage profile, which is a clear indication of genomic rearrangements. But the precise composition of the integration site, or multiple sites, is not resolved with TLA due to short read sequencing. Nanopore sequencing generates long reads compared to TLA, and these reads become handy for resolving the integration site structure and vector concatemerization. I have already showed this table before, saying that using the short reads we see only the vector connections and can't reconstruct the order of the vector copies in a concatemer, because reads don't span from one copy to another. But knowing the integration sites, we can design guide RNAs on the genome next to the integration site, and from there start generating long reads spanning the entire integration locus, if it's not too large, of course. Then in many cases, the integration site can be reconstructed. For example, in this case, it was shown that 8 pieces of the vector fragments were connected to each other in different orientations. Some are really small fragments, maybe not even containing the entire expression cassette. On top of that, it was also shown that the chromosome 8 was connected to chromosome 5, so it's a genomic translocation. This data can explain the atypical coverage profile obtained with TLA. Additionally, such sequencing can also give you the insight about which integration site contain the sequence or structural variants of your vector. As you can see, using just a few assays, you can get a very detailed data characterizing your line. And you might ask, do I really need so much information at each time point of the cell line development? In this final slide, I would like to go back to the steps of cell line development and indicate what do we suggest to our customers to do at each step. For example, for vector sequencing, no TLA or nanopore is needed. The vector itself can be just sequenced with NGS. Then, different transfection methods can be tested with TLA. The goal here is to describe the vector integrity and integration sites, because, for example, in a system where you use random integration, you might get one to several integration sites with one or multiple vector copies per integration. But if you're using transposome-based vectors, then you might get up to 20-30 integrations. Based on the integration profile, you can choose the method you want to stick with. Then, when that is done, you might obtain a lot of clones, which we can quickly screen with TLA, only describing the mutations in the vector and the independent nature of the clones based on their integration sites. That information helps you to choose the best clones. And if you want to dive more into the genetic organization of the integration sites and vector concatemers, the long-read nanopore can be added for a more complete picture. And then, when you do your cell banking, master cell bank has to be characterized, and for that, we combine TLA with ddPCR, because copy number estimation is one of the requirements for the submissions. If you happen not to have a picture of that single cell that gave rise to your MCB, and you still have to prove monoclonality, it's possible to do that on a genetic level as well. For that, we again use TLA data of your MCB and also ask you to provide us with 93 subclones of the bank. Why is that? The breakpoint sequences identified with TLA, for example, the integration site breakpoints, are very specific characteristic of your line. They will not be present anywhere else. We then test 93 subclones for the presence of a selected breakpoint. That is done with a qPCR. If all 93 subclones are positive for the same breakpoint as MCB, it means that they are all really the same. Using the published statistical test, the experiment shows that your MCB was originally monoclonal. And the last but not least, it's also important to show genetic stability of the line. That at the end of culturing or at any other stage in between, nothing has changed in your cell line. For that, we also perform TLA and ddPCR, but now on both cell populations, and compare them between each other. If nothing changes, then the line is stable. I hope this overview will be useful for decision-making on cell line testing at different stages of development, because at the end, we all aim to develop the safe and efficient drug products for the patients. Thank you for watching.
Thank you, Irina, for the excellent presentation. And of course, thank you to everyone who joined us today. We're now at the end of the session. While we won't be able to answer questions live today, we are collecting all questions submitted in the Q&A box, and our team will follow up to respond directly. Once again, thank you for joining us today. We hope you found this session valuable, and we look forward to connecting with you soon.